Hello everyone, welcome to Get My Class. So let's solve this question which says the foreign DNA is inserted into the fragment that serves tetracycline resistance in PBR322. So which of the following observations is true? Option A, recombinants will not grow on ampicillin containing media and tetracycline containing media. Option B, recombinants will grow on tetracycline containing media. Option C, recombinants will grow on tetracycline containing media but not on ampicillin containing media. Option D, recombinants will grow on ampicillin containing media but not on tetracycline containing media. So let us understand the question. The question says the foreign DNA is inserted into the fragments that serves tetracycline resistance in PBR322. So which of the following observations is true? So what are they asking? They're saying that when the foreign DNA is inserted into the fragment that serves, that is tetracycle resistance in PBR322. So what will be the observation? What will be the result? So before we look for the option and further discuss about it, first of all we have to know what is this PBR322. Of course, it is a type of a vector. Okay, it is a cloning vector into which the foreign DNA or gene of interest can be inserted. Okay, it can carry the gene of interest. Okay, and further it will be transferred into the host, and inside the host this vector will express itself like the gene of interest along with the other genes whichever are present on the vector will get expressed. So expression of genes, or you can say recombinant DNA, happens in the host. Okay, now when we say PBR322, it is actually a plasmid, P for plasmid. Okay, BR322. So it is a plasmid. Here they're talking about insertion of the foreign DNA in the tetracycline resistant gene. So it has two selectable markers. One is called as the TET-R, that is tetracycline resistant gene, and the second one is called as AMR, that is ampicillin resistant gene. These are the two major selectable markers which are present in the plasmid, that is PBR322. So the function of TET-R is to, whichever organism is having this plasmid with this gene, it will be resistant to, that organism will be resistant to tetracycline. Okay. That means here, if the organism is having this gene and in active form, then it will be resistant to tetracycline. So that means it will grow on tetracycline containing medium. And also here, if it AMP resistant gene is, if it is active in nature, like it is not yet inactivated, it is complete. Okay, that will give resistance to, here the organism will be resistant to ampicillin. Okay, what is ampicillin and tetracycline? These are two different types of antibiotics which can actually kill if these genes are not present or if these genes are inactivated. So here they are saying that the foreign DNA has been inserted within the tetracycline gene. Suppose you have the foreign DNA, that is the gene of interest, you have inserted within the tetracycline gene here. Okay, so what will happen? This tetracycline gene will get inactivated. So this is your DNA, suppose foreign DNA. Okay, that when you try to insert within the tetracycline gene, this will get inactivated. So when it gets inactivated, what will happen? It will lose its resistant power towards tetracycline. Okay. Now it carries the foreign DNA, that is fine, but the tetracycline resistant gene has been inactivated. So now this recombinant DNA, which is carrying our gene of interest, when this is introduced into the host, what will happen? This host will be resistant to ampicillin. It will be having active ampicillin resistant gene, whereas tetracycline it will not be resistant. So the recombinant will be able to grow in ampicillin ampicillin containing medium, because it still has its ampicillin resistant gene intact. Whereas in tetracycline medium, it will not grow. Okay, because the tetracycline antibiotic is there, and here the gene for resistance is inactivated because of the insertion of the foreign DNA. So this insertional inactivation, this is called as insertional inactivation, because of insertion that the tetracycline gene is getting inactivated. The tetracycline resistant gene is getting inactivated. So let us see. Option A says recombinants will not grow on ampicillin. No, recombinants will grow in ampicillin because ampicillin gene is fine, so it will grow in ampicillin and tetracycline containing medium. It will not grow in tetracycline, that is correct, but not in ampicillin. Ampicillin it will grow because it has resistant gene, hence it is similar. Option B says recombinants will grow on tetracycline containing medium. No, because the gene for tetracycline has been inactivated because of insertion, hence it is ruled out. Option C says recombinants will grow on tetracycline containing medium but not on ampicillin containing medium, which is again ruled out because, as I said, tetracycline gene has been inactivated, so the resistant gene has been inactivated. Then how will it grow on the medium containing tetracycline? So it is totally ruled out. Option D says recombinants will grow on ampicillin. Yes, because their ampicillin resistant gene is intact, but not on tetracycline containing medium. That is absolutely right because tetracycline gene has been inactivated by the insertion of the foreign DNA. Hence, that is the correct answer. So let us conclude. The answer is option D. I hope you understood the explanation. Thank you.